Mission failed. We'll get them next time.
No! God, please, no!
For this, we're going to go over a quick overview of kind of the differences between PCR and Sanger sequencing. So let's take this plasmid, for example, this PJAK19, where the top strand and the bottom strand are both labeled accordingly. Now, if we wanted to do PCR on this gene right here, let's just say it's this awesome gene, it's 1492 base pairs long. What we need is two sets of primers for PCR. So we need a forward primer, which anneals with the bottom strand, and then a reverse primer, which anneals to the top strand. So to start our PCR, we are going to start with the template, and just to make things clear, I've changed it so it's not a circular template, it's just represented as a linear DNA right now, but don't forget that it is still circular, so it will go around and come back to itself, basically on each side. And the gene that we are trying to do PCR on is represented by these black bars right here. So the first step in PCR is denature. So we're going to go ahead and denature, and the two template strands will come apart. And then the next step is annealing. So this is where our primers are going to anneal or base pair to their complementary primer regions on the template strand. Then in extension, we are going to get the extension, and the primers will extend out and make a new strand of DNA. Now, one thing to remember, though, is that the Taq polymerase doesn't know where to stop. So it will keep going until it gets to the end of a strand of the template's DNA. Now, because there is no end on the template DNA, since they're circular, it will keep going around. So the only thing that's stopping it is the time of the extension. So it's not going to go all the way around the plasmid, but it will probably far exceed the gene that we are actually trying to do PCR on. So in this first round, any product from it is not going to be the correct length. It's going to be overextended. And we're not going to be getting correct length product until later steps of PCR, which we will show. So in order to go to the next step, we'll take these and denature them. And essentially any new strands that have been made, instead of having the dashes, they're now represented just by solid lines. Again, though, they extend out past the gene that we are actually trying to do PCR on. So we'll take these to the next step, to where we will anneal our primers and then extend out. So now in this step, on this top strand right here, again, we have the primer and it's being overextended. So this is longer than what we want as our final product. Now in the second one here, this primer went this way, and then it was met with this end. So this is actually the end of this strand of DNA. Remember, this one is not circular, this is linear, and this is the end of it, and it corresponds with the end of our gene that we're going. So this new DNA strand on the top right here, this is of the correct length that we are looking for. So this is the correct length PCR product. And again, so it's mirrored in these bottom versions here, where we have this direction, primer extension, which again, it gets stopped right here because there's no more DNA on the template. So this one, this top strand is again the correct length. But then on the bottom here, where we are extending off our original template, remember this is circular. So this will go on and this is going to overextend. So we're going to have incorrect length product. So now we will go to the third step. So we'll take all these and denature them. They're all going to come apart. And then we can do the annealing step. So all the primers are going to go on. And then we will do the extension. So we're going to extend. All right, so what's happening here? For clarity, I'm going to throw up these dotted lines here. And what these represent, so this is the essentially the range of the gene that we want. So anything that is bound within these bars, any product, that is correct length product. Anything that has gone past them, though, it's extended, it's too far extended, it's not our full length product. So again, you can kind of go down and see now we are starting to generate a good amount of the correct length product. Here we have a new one, here we have a new one, here we have a new one, here, here, and here. Okay. And in terms of generating overlength product, again, it's only going to be from our original templates here on the bottom and then on the top. So as PCR continues, remember, this original template, it's still in the mix. So it's always going to be generating a little bit of extra length PCR product that is going to be no use to us. But what the more important thing is that as the rounds continue, the amount of correct length PCR product is going to exponentially increase. So now let's take a look at Sanger sequencing and how it's a little bit different than PCR. So again, we are going to start with this is still the circular plasmid DNA, but in Sanger sequencing, we only have a single primer. So all we're doing is we're just making a copy of one strand of this. So we'll take the top strand, for example. And again, remember in Sanger sequencing, you're going to be using a lot more of the template than you would be using in PCR. PCR is for amplifying a template, whereas Sanger sequencing is you're sequencing it. And again, in the cycles in Sanger sequencing, we're only getting linear growth. It's not exponential. So we need a lot of template around in order to actually get good results from the sequencing. So we will have our primers come, and they're all going to anneal in the same spot. And then what happens is that, let's just say, for example, this is the reaction mix that has the DD ATP, for example. So it has the dideoxy ATP. So anywhere there's a T on the template strand, it could potentially stop the growth of the new strand because it can go put in a DD ATP and that will stop growth. So we'll just run a little simulation here just to see. So the Taq polymerase will come and then start to extend, and it's going to make all these different length products. And one thing to remember is that in the Sanger sequencing that we did, the primer itself actually had the fluorescent label on it. So you can see right there, it's on the 5' end of the primer. So we're going to take our sample and then load them all up on the gel. And then as the gel runs, they're going to separate out into lower bands. So down here at the bottom, this was the shorter length product. So this is the ones where it terminated a lot earlier, kind of in the beginning of the sequence. Whereas up here, these are the longer length ones. So the ones where it went a lot further. And you get this separation here. And this is just example of one nucleotide. You would have to run all 4 and then kind of build backwards, start from the bottom and start to essentially assign nucleobases all the way up to the top, building it from 5' end of your primer all the way to the 3' end extension. So just as a little practice here, this is just an image of a gel that I made, and each lane here, so we have the A lane, the C, G, and the T. And so just for practice, you can try to figure out what the sequence of the Sanger sequencing results are, and then what the sequence then of the original template strand was. So you can pause the video now to kind of look and write out the sequence. To get the sequence from the Sanger sequencing product, again, you're going to start at the bottom because this is essentially synonymous to the 5' end, and then you're going to work your way up. So here we can see we have 2 C's. It goes like C, C, and then there's a T over here, and then we keep moving up a little bit, and then there's an A, C, A, G, A, and so on. We just keep moving up, and then this is the resulting sequence from the Sanger sequencing. But remember, this is a reverse complement of your original template. So the original template that was sequenced would actually be this. So it's the reverse complement where the 5' end is over here. So the template that we originally sequenced, it was A, C, T, C, C, T, and so on. In order to do sequencing on DNA, you are first going to need to know something called the ambiguity codes of nucleotides. So we have our 4 basic nucleotides, A, C, G, and T. And this is good if you know exactly that it's that nucleotide whenever you're doing the sequencing. But gels do not run perfectly. So there might be instances where there might be two bands, say like an A and a C, that kind of line up and their peaks really line up, and you don't necessarily know exactly which one to call. And you don't want to pick one or the other. So we need some nomenclature in order to denote if we want to represent either possible 2, 3, or even 4 nucleotides at a certain position. So this is the nomenclature for that. So we'll start off with a situation where it could be either A or T. Now both of these have 2 hydrogen bonds that they have with each other, so we denote them with W for weak. Next we have C and G. These both have 3 hydrogen bonds between one another, so we denote them with S for strong. The next combination is A and C, and we use M for amino because they have amino groups at the top of them. Next is G and T, and these are denoted with a K for keto because they have ketone groups on the top. We then have A and G. These are both purines, so we use R for purine. And then C and T are pyrimidines, so we use Y for pyrimidine. So now we get into the situations where there's 3 possible nucleotides. And so in this case, we have C, G, and T. So it's not A, so it's the next letter in the alphabet, which is B. And then we can have A, G, and T, so it's not C, so it's D. And then following the trend, we have A, C, or T. It's not G, so it's H. And then last, we have A, C, and G. It's not T, so it's U. Oh my gosh, it's uracil. I totally forgot about you. Oh, what? He said, brah, you can't forget about me. I know I don't have a methyl group like diamine does, but I am still important. That is entirely true. Uracil is extremely important in RNA. So we can change this here. So we'll do not T, so V. Okay, good. And then we might have a case where it's any one of the 4 nucleotides. So with this, we just denote it with N for nucleotide. You're going to want to open up the internet and search for 4-peaks, and then download this first one by Nucleobytes. Let's go ahead and click on Get It for Free, and download the program. Once the program opens, you're going to want to open up one of the data files. So we'll go ahead and we're going to pull one of them and open it up. So usually I like to start by dragging the slider over just to make the peaks a little bit wider so it's not too confusing to look at with a bunch of sharp peaks just right on top of one another. So you'll notice when looking through your results that the sequencing program did a lot of the work for you already. It took the chromatographic results and it called a lot of the bases already. Especially towards the beginning and middle of the sequence, a lot of the bases are going to be called correctly because the data looks good. Now, as you start to go further and further within the data, we know that the sequencing gel loses sensitivity, and the peaks are not going to be as defined, they're not going to be as sharp, so the program might have difficulties calling some of these nucleobases. So especially towards the end, you might get things like the ambiguity codes, or even things where it uses a lowercase letter instead of an uppercase letter. In the case of a lowercase letter, what that means is that the program isn't quite 100% sure that it's supposed to be, but it's sort of sure. So it kind of leaves it up to you to make the final call. And then for the ambiguity calls, you can either put those in, or you can confirm that the sequencing program made the correct call on it. So in this example here, I'm showing what to do in case you need to change a base that was called. So what you're going to do is you're just going to double click on the letter up there, and then you can change it to whatever you want. So you can do this for the lowercase letters if you look at the chromatogram and it looks okay. and you feel confident in calling that, you can change that to a capital letter to denote that it's confirmed that that is it. Additionally, if there's an ambiguity code that you feel can be resolved or that needs to be added, again, all you need to do is just double click on the letter and you can type in what you want. Alternatively, if you do select a letter, you can then just click on delete. If it called something where there wasn't actually a peak there, you can just go ahead and delete that letter. You can also insert letters by double clicking on a space in between two letters, and that will insert a letter there. When you are all done analyzing your data and you're ready to do a blast search, what you're going to do is come down and select this cog and then click on blast sequence and then just do default. It's the easiest one. And what this is going to do, it will automatically open web browser and do the NCBI blast search with all the parameters already put in. So all you have to do is just scroll down and then click on blast and wait for the results to come up. When the blast search results finally come up, you'll be able to see all of the hits that your sequence generated. And what you are tasked to do is to find out what gene you actually sequenced. So all of these plasmids here, or DNA, have that gene within them. And so you need to figure out what that gene is. Now, I would say try to look for something common amongst all of these names here. One thing to point out, though, is that plasmids are typically, or almost always, named by putting a lowercase p at the beginning of the name, and then some letters or numbers to denote what the plasmid actually is. But you are looking for a specific gene rather than a plasmid itself. So the gene is in some of these names, but more than likely, if you have something that's repetitive, like what's seen right here, this whole list of things where just a single letter or number changes, that's just describing a plasmid and not necessarily the gene that's in the plasmid. So try to look for other ones and see what you can actually find. If you do want to explore, you can also click on one of these hits and it will show you how your search query actually lined up with the subject query. So essentially what's on the top row of all of these is the sequence that you submitted, whereas what's on the bottom row is what was deposited into the database. So you can see how they actually line up, where the sequences align, where there's mismatches. Again, these might just be slight variations in the gene, mainly mutations in the protein, small little mutations and whatnot. But we can see that in this one here, there is a lot of matches from what we submitted to what was actually on the database. One thing you are going to have to do with your data after you found out what the gene that you sequenced is find the amino acid sequence actually in your data. So the amino acids are denoted by the little gray letters that are right below the nucleic acid sequence. And one thing you are going to need to try to do is manipulate the frame shift of it. So that's going to be right down here, and you can click on frame +1, +2, or +3. And what this is going to do is it's going to change what the first nucleotide in the codon sequence is going to be. So what this defaults to and assumes is that the first nucleotide that you have is the first letter of the first codon of your amino acid sequence. Now, our sequencing isn't perfect. We don't necessarily know where we started to sequence, where we started to get good data. So we don't know that that's necessarily true, whether or not the first nucleotide is actually the first, second, or third one within its respective codon. So what we need to do is to change the frame shift, and whenever you do change the frame shift, that will change what the subsequent amino acids presented are. So you are going to need to maybe play around with this to find which frame shift is correct for getting the correct amino acid sequence for the gene that we sequenced. Once you have found the correct frame shift, you're going to need to copy and paste the translation sequence. So you can click on this little info right here and then click on translation, and this is just a text basically of the amino acid sequence. So you can just go ahead and copy and paste this into a Word document for your assignment. Now, because we did have two different primer sets for this, if you were looking for your translational data and you went through all three frame shifts and you still couldn't find the correct one, that probably means that you have the primer that's going in the other direction. So what you're going to need to do is you're going to need to flip your data. And what you can do is you can click on this cog and then click on flip. And what this will do is it will transform the data that you have to the reverse complementary sequence. With that, then you can redo the frame shifts to actually find the correct translational sequence. So the first thing you're going to want to do is open up a web browser and search for Chromas Lite. And then you're going to click on this Technolysium one right here. And then you're going to go ahead and download the program. So when you have the program open, you're going to go ahead and click on this Open tab right here and select which sequencing data file that you want to open up. You'll notice when looking through your results that the sequencing program did a lot of the work for you already. It took the chromatographic results and it called a lot of the bases already. Especially towards the beginning and middle of the sequence, a lot of the bases are going to be called correctly because the data looks good. Now, as you start to go further and further within the data, we know that the sequencing gel loses sensitivity, and the peaks are not going to be as defined, they're not going to be as sharp, so the program might have difficulties calling some of these nucleobases. So especially towards the end, you might get things like the ambiguity codes, or even things where it uses a lowercase letter instead of an uppercase letter. In the case of a lowercase letter, what that means is that the program isn't quite 100% sure that it's supposed to be, but it's sort of sure. So it kind of leaves it up to you to make the final call. In order to change a lowercase letter to an uppercase, all you're going to do is just double click on this, and then you can go ahead and type in an uppercase letter right there. Alternatively, if you find a base that you feel shouldn't actually be there, like the program called something that wasn't actually a peak, you can go ahead and highlight that. So just click on it and then press on delete, and that will just remove that base from being called. You can also insert letters by double clicking on a space in between two letters, and that will insert a letter there. What you need to do to analyze your data is to go through and just make sure that the program did the correct job in terms of calling the bases. Towards the end of the sequencing mainly is where a lot of problems arise. So what you need to do is just make sure that, one, that it called the correct bases, two, there's any lowercase letters can be changed to uppercase letters, and then the addition of any ambiguity codes or any insertion or deletion of bases. Once you are done analyzing results, we can go ahead and do a blast search by clicking on File and then Blast Search. It will put all the parameters in. Don't change any of these. Just go ahead and click on Send, and then it will automatically open up the NCBI website in your web browser. So you can just click on View Report, and that will show all of the hits that you generated from your sequence. And what you are tasked to do is to find out what gene you actually sequenced. So all of these plasmids here, or DNA, have that gene within them. And so you need to figure out what that gene is. Now, I would say try to look for something common amongst all of these names here. One thing to point out, though, is that plasmids are typically, or almost always, named by putting a lowercase p at the beginning of the name, and then some letters or numbers to denote what the plasmid actually is. But you are looking for a specific gene rather than a plasmid itself. So the gene is in some of these names, but more than likely, if you have something that's repetitive, like what's seen right here, this whole list of things where just a single letter or number changes, that's just describing the plasmid and not necessarily the gene that's in the plasmid. So try to look for other ones and see what you can actually find. If you do want to explore, you can also click on one of these hits and it will show you how your search query actually lined up with the subject query. So essentially what's on the top row of all of these is the sequence that you submitted, whereas what's on the bottom row is what was deposited into the database. So you can see how they actually line up, where the sequences align, where there's mismatches. Again, these might just be slight variations in the gene, mainly mutations in the protein, small little mutations and whatnot. But we can see that in this one here, there is a lot of matches from what we submitted to what was actually on the database. Once you have found out the protein for the gene that we sequenced, you are going to need to find the translational data or the amino acid code within your sequence. Now, in order to do that, in Chromas, it's not normally displayed. So what you're going to do is you're going to first click on Options and then Translational Data, and then you can select either frame +1, +2, or +3. And what this is, this corresponds to the frame shift of the amino acid sequence. So we know that the nucleotides, whenever they get translated into an amino acid, it's a codon, or 3 nucleotides in a row, that determines what amino acid will be there. Now, whenever we are doing our sequencing, we don't necessarily know where our sequencing starts, or more importantly, where we start to get good data. So, for example, the first nucleotide in our sequence, we don't know if it corresponds to the first, second, or third nucleotide in its respective codon. So what we need to do is to figure that out. So you need to go through the different frame shifts, and each one is going to give you a different sequence of amino acids because it's going to dictate where the codon starts and stops. So you are going to need to go through the different frame shifts and find the one that actually produces the correct translational data or amino acid sequence, and find that for your homework assignment. In order to actually copy and paste the amino acid sequence, what you're going to do is you're going to need to click on Edit and then Copy Translation, and then you can select either frame 1, 2, or 3. So whenever you select it, that will just put it into your clipboard essentially, and you can just go into a Word document that you're doing your assignment on and paste the sequence into that. Now, because we did have 2 different primer sets for this, if you were looking for your translational data and you went through all 3 frame shifts and you still couldn't find the correct one, that probably means that you have the primer that's going in the other direction. So what you're going to need to do is you're going to need to flip your data. To do this, you're going to go ahead and click on this reverse button up here. And what this will do is it's going to flip your data so it's the reverse complement of whatever you sequenced. And then with this information, you can re-look at the frame shift data and the amino acid sequence to actually find the correct order amino acid sequence for the protein that we're looking at.